afternoon to everyone. The pessimist sees difficulty in every opportunity. The optimist sees the opportunity in every difficulty. I am Dr. Nitya Prabhaharan, Assistant Professor, PG and Research Department of Microbiology, to marshal the program. I feel honored to invite all the dignitaries, participants, and partakers taking a keen part in the important program. I'm sure that you will feel enriched with knowledge after completion of today's event. Again, it is a glorious moment to extend my warm wishes to the International Faculty Development Program and hope that you will have a great time ahead. Now, I invite Dr. R. Krishnaveni ma'am, HOD, Organizing Chair, to start the third day session of International Faculty Development Program.
Good evening to all. I invite you all today to attend the presentation by Dr. Kasi Gopinath, Postdoctoral Fellow, School of Materials and Energy, Southwest University, China. In the title of Recent Trends in Green Nanomaterials used for plant development and biomedical applications. Now I invite Mrs. T. Meghala ma'am to deliver welcome address and introduce the guest.
A meaningful and warm good afternoon to all. According to Benjamin Franklin, one today is worth two tomorrows. What I am to be, I am now beginning. Yes, today is the pleasant day in the presence of God Almighty. It gives me immense pleasure to deliver the welcome address for today's session, Recent Trends in Green Nanomaterials Use for Plant Development and Biomedical Application in International Faculty Development Program organized by PG and Research Department of Microbiology, Idhaya College for Women, Kumbakonam. Leadership is the ability to get extraordinary achievement from ordinary people. I would like to welcome our principal, Reverend Sister Dr. Yuji Namala, who inspires us as a leader to achieve extraordinary things. Leaders are not made, they are born. I welcome our secretary, Reverend Sister Claudine Arokya Mary, who sculpted herself as a leader. A supporter finds potential in people and processes them. I would like to welcome our administrator, Reverend Sister Andoniamal, and Vice Principal, Reverend Sister Dr. Vimali Mary, who supports us for all our efforts. Work hard in silence. Let your success be the noise. I am delightful to welcome and introduce our successful resource person, Kasi Gopinath, MSc, MPhil, PhD, Postdoctoral Fellow, School of Materials and Energy, Southwest University, Changping from China. He has graduated in Nanoscience and Technology discipline from Arakappa University in 2016. Previously, he was working as a postdoctoral fellow during 2017 to 19 under Professor John Chul Seo at Department of Packaging, College of Science and Technology, Yonsei University, South Korea. The primary focus of the research is the design and development of low-cost, efficient, long-life materials that can facilitate widespread commercialization of polyurethane, nanocomposite coatings, and for biomedical applications. My deepest welcome to you, sir. It is my pleasure to welcome the participants, whom includes professors, associate professors, assistant professors, research scholars, and students of various disciplines from various universities and colleges. The more you learn, the more self-confidence you will earn. As per the words of Brian Tracy, once again, I invite you all to replenish your knowledge through this international FDP. Thank you.
Thank you, ma'am. Thank you very much for your wonderful invitation. Now, I hand over the session to our resource person, Dr. Kasi Gopinath, sir. Sir, now the session is yours.
Okay, thank you, madam.
Thank you, sir.
Good evening to one and all. Madam, are you audible?
Audible, sir. Sir, shall I
Okay.
share the screen, sir?
Oh, just a minute. Okay,
Okay,
I'll
sir.
tell
Okay.
you. Good evening to one and all. Thank you for the short and smart introduction. First of all, I would like to thank for the management of Idhaya College for Women's Kumbakonam. Secondly, I sincerely thank convener and organizing chair, Dr. N. Krishnaveni, assistant professor and head, Department of Microbiology, invited to me the International Faculty Development Program on Biological and Industrial Application. And I thank to organizing committee, Department of Microbiology and dear participants. Now we are going to the presentation. Today my topic, madam, show me the slide. Excuse me, ma'am.
Gobinath, sir. Sir, you can continue, sir.
Please show me the slide. Presentation.
Okay, sir.
Hello, Nitya.
Ma'am, just I share it, ma'am. Share it, ma'am. Just a few minutes, ma'am. Sir,
Sir?
it is visible.
Yes, ma'am. Not.
Sir?
Cannot. Okay, madam. Just a moment. Okay. Okay, today's topic is recent trend in green nanomaterial used for plant development and biological application. In myself, Dr. Kasi Gopinath, School of Material and Energy, Southwest University, Chongqing, Hebei, China. Today, nanotechnology. Nanotechnology is booming field and multidisciplinary application in frontier research. Nanotechnology is compressing in the 1 to 100 nanometer of the object scale. The 1 nanometer is 1 billion in 1 to 10 to the power of 9 meters. So nanoparticle is studying about 1 to 100 nanometers. The nanoscale. Nanomaterials. What is different between the nanoparticle and nanomaterials? Nanoparticle is 1 to 100 nanometer. More than 100 nanometer is called as a nanomaterial. Nanocomposite, more than 1 or 2 material as considered as nanocomposite. It can be used to biological application. Nowadays, nanomaterials being employing the plenty of application due to the excellent physiochemical properties, depends upon the size and shape. It's a nanoparticle have a very good unique property is high surface to volume ratio. Nanotechnology is applied for, now next slide. Second one. Just okay. Next. Okay. Correct. Application of nanotechnology, different field: medicinal, drug, energy for dye solar cell, medicine for drug delivery, and nano biotechnological applications, and nano device and optical energy application, and defense and bioenergy, and cosmetic and nanofibers, and biosensor and removal of pollutants. Next. Outline of our theme is consisted of six categories. First one is introduction. Second one is synthesis and characterization of metal and metal oxide nanoparticle used for the biological application. Third one is the how to improve the plant development. Third one is used for pollen germination and mitotic cell division. Fifth chapter is considered for cytosol nanoparticle preparation and used for the plant regenerative application and plant growth promoters. Last one is the metal doped metal oxide nanoparticle used for plant growth development. Introduction. The importance of medicinal plants. India have a lot of medicinal plants, more than 200 species used for the medical application. Is medicinal application is used for three medicinal uses: Ayurveda, Siddha, Unani system of medicines. The World Organization has been reported in the large group of people or depends on the conventional medicines, the physiological and physical health requirement. The particular advantage of traditional medicine is cost-effective, non-toxic, and modern treatment. They are Indian traditional system of medicine considered of the Siddha, Ayurveda, Unani. Siddha is Tamil traditional medicine, is more than 16 Siddhas is called as Agatyar and Boger and Kumbakonam. There are so many, 16, 18 members is more important. And the Ayurveda is Indian subcontinental, is in subcontinental medicines, is medicine. Unani is the Arabic traditional medicine. Arabic traditional medicine is the empire of, it is Mughal Empire, introduced by Mughal Empire. There's so many medicinal plant used in the three system is nowadays called as Ayush. In Siddha peoples more than Siddha as mentioned, 4,448 diseases are cured by medicinal source. Already you know the Palani temple, the statue made of Bogar mentioned lot of metal and metal composite is called as arsenic trichloride, arsenic trioxide, arsenic dichloride, and arsenic pentasulfide, and sulfur and mercury. So the unidentified component are in the statues. And Ayurveda is mentioned, the Tanvantri is mentioned, the Sanskrita books told several medicinal plants used for medical used for the medicinal uses. Last Unani is European medicine, Greek and European medicine is introduced by the Mughal Empire. The old traditional peoples have the very good knowledge about the metals. The first invention is copper. Over 5,000 years ago, Egyptian, Greek, and Persian Romans have stored the food in silver brass vessels. Similarly, Asians, Indians are silver, gold, copper-based jewels is Bengal, Bangladesh, Anglet, and as well as drinking water use the kitchen vessels. The metals are good antimicrobial property. You know one thing, in the child people used for the anglet in silver. Why people use this silver in the anglet? When in the childhood stage, and the child have the fecal matters or stools inside of the body, is microbial contamination. So in order to avoid the microbial contamination, used for silver anglet. And gold and belt, silver belt also used for in order to avoid the microbial contaminations. If you keep in the water in the copper vessels, if you easy to copper vessel, all the microbial are killed in 24 hours. And the other thing is the copper is easy to wash to material and easy to wash to material with the help of acid medium. We can use the turmeric cake or lemon pots. You can use the easy to wash material. Furthermore, Asian medicine, Ayurveda used powder form of copper, iron, silver, and gold, and various formulations. Formulation in the shift of metal vessels and abnormal metal powder ancient was shifted to the bulk level to nano size. The traditional system of medicine rooted knowledge from the macro level to nano level. Drug carrier for that around 2000 BC. Also the honey used for drug carrier. The presentation of nanomedical plant-based extract whereas used for including the capping purpose. The samples are calcinated. Calcinated means heated and 300 degree, 200 degree, 1000 degree, 500 degree. Get a powder. The formulation used for drug carrier and treatment of disease and cancer and skin infections. So okay, whatever our theme, the importance of three plants, and the Kuruji part, the Kabilar mentioned the 99 flowering plants. We have choose of three plants. Kanthal is glorious or superpa, is also called as a Kanvali Poo. Another one is Termeli Arjuna, is called as Neer Marudu or Well Marudu, the Mardam plant. Termeli Arjuna. Last one is Karandai. Karandai is called as Pyranthus indicus. This is a herb. This is a Termeli Arjuna is a tree, and Glorious Superpa is a tendril herb. The Gloriosa Superpa is a glorious means is a happiness. Gloriosa Superpa is old and is belong to the Liace family. Currently, it's considered as a Colchicinaceae family. Climbing herb, natively south. Okay, next one, correct, madam. Native in South Africa, Glorious tuberous plants with a blow shape like the V or L shape like is used for the skin disease, leprosy, snake bite, and bleeding of biles, and so on. So, the tuberous seeds contain the two alkaloid, major alkaloid, colchicine and colchicoside. Everyone know that colchicine is the alkaloid is used for the polyploidy. Polyploidy means this doubling of the chromosome without the cell division, the arresting of the inactivate thus release for formation. So you can use it for the favorable things for plant development. And leaves contains some of the gloriosine, colchicine, and gloriosine, and so other component is there. In another thing, why these plants These plants, more than 17 countries issued by the sample post because of Uganda, Malaysia, and so many countries are issued by the postal stamp. In India, 1977 issued the Indian stamp also. Why the plants are more important? These plants is the national flower of Zimbabwe, state flower of Tamil Nadu, and national flower of Tamil even. Nowadays, Tamil is not there. Is the Mavira days are celebrated November 27th. And another thing is more abundant in Prosopis julifera and palm trees. Is cultivated. Next time, madam. Is cultivated. Is plants widely. Hold on this slide, please. These plants widely cultivated Ariyalur, Dindigul, Erode, Karur, Namakkal, and Nagapattinam, Tirupur, and Tiruchi districts, and as overly cultivated in Mulanur, Vattachitram, Aravangai, and Ampegi, and Tarapuram, so Attur, and Jayakonda district also. This medicinal component. This medicinal component of colchicine is very tradable medicine. So Italy is one of the largest superba seed buyer, and because of the huge demand of medicine in European countries, it is the 1 kg of this plant seeds is 30 years ago 250 rupees. Now 3,430 rupees is currently market price. Just a moment. Okay. Last one. Next one, Terminalia Arjuna. Terminalia is a competition family, large evergreen tree, 30 to 30 meters. Tree is all over in Burma, India, and Sri Lanka. This bark extract contain a lot of phytoactive component, especially for tonic acid, argonic acid, and toluene derived component. Leaf extract contain leucoanthocyanin. The extract contains the fruit extract contains safranin, flavulin, glycoside, polyphenol component. This is the argonic acid structures and phytocomponent. And next one, Phlerantha indicus is called as the Kotang Garandai. Is a gracious, as everyone know. After the harvesting of the paddy field, you can see this plant. It is a very good, important medicinal plant. It traditionally used for anti-HIV, antiviral property and anti-HIV applications. Also, it's very good, important medicinal plant. Why we choose the plant tissue culture? Plant tissue culture is important for our alternative tool for regenerated plant. In the tissue culture is using for tissue and fragment and calais and isolated cell. The commercial aspect of the tissue culture form referred to the micropropagation. The one of the techniques for tissue culture, micropropagation referred to the propagation of whole plant. The tissue culture derived from the explant is a unique clonal propagation. So everything is same germplasm. The last one, nanoparticles. Now I will continue to the topic, irrelevant to the topic. Nanoparticles are important plant growth of the Justica, latex saliva, and Biomedical and cucumber saliva. TiO2 zinc oxide and aluminum oxide nanoparticle was synthesized. AU and silver and nanoparticle seed germination has a positive impact on the plant vegetative growth of Arabidopsis thaliana and Brassica gentiana. Next, because of AU nanoparticles is a metal regulating protein for protein synthesis. The introduction of metal and metal oxide nanoparticles, silver, gold, or metal, ruthenium or metal nanoparticle is the application for drug carrier and imaging biosensor application. Ruthenium oxide nanoparticle also used for pollutant and azotide degradation also. The cerium oxide is metals are used for biosensor application of dopamine biosensor and so on, and biosensor and solar cell application. Methods of nanoparticle synthesis. Generally, people are using three methods: physical method, chemical method, biological method. Biological method divided to the phytosynthesis and medical microbial synthesis. The physical method is a physical vapor deposition, microwave assisted, and so many methods of physical method. Chemical methods are solvothermal method and co-precipitation method. Biological method, we are using the plants derived the different parts: leaf, stem, root, tubers, and so on. So different part used for the phytosynthesis. Microbial, intercellular, and extracellular. Generally, we are used to the extracellular component used for the reduction process. Why the chemical component and the energy physical methods are used expensive energy consuming process. The chemical methods are some of the hazardous: nuclear formaldehyde, dimethyl formaldehyde, triethylamide, and hydrogen hydrate using hydrogen is causing some hypoglycemia and neurodegenerative changes and hypotension toxicity and cancer. Dimethyl formaldehyde produce the stomach aches and vomiting. Triethylamine as used for as make as a irradiation skin, eyes, mucous membrane. So the chemical component are very toxic. So in order to avoid this manner, we want to choose biological method. Biological method is cost effective, eco-friendly, easy to scale up the large production, and also no need to use any extra pressure, energy, and temperature and toxic chemicals. Previously, some of the reports are available in the synthesis of nanoparticle. The aloe vera, azadeta, indica, implica, application, jatropha, curcum, musa, paradisiaca, and Pajum, Pesidium, Goagium, Avia brasiensis, Avina saliva, coriander saliva. So the plants are used for already synthesis for silver, gold, platinum, palladium, nanoparticle, as metal oxide for zinc oxide, copper oxide, cerium oxide, titanium oxide, nanoparticle. It shows the very good antimicrobial property. The antimicrobial property against Escherichia coli, Bacillus subtilis, Vibrio, cholera, Pseudomonas aeruginosa, and Staphylococcus aureus. And also the anti-biofilm activity for nanoparticle is incorporated into the polymer matrix. It shows the very good coating material for biological application in plants, implantable material, and also look like a polyurethane is a biopolymer. Is you can use for the metal nanoparticle, metal and metal oxide. You can use the avoided biofilm formations. And another thing is the inorganic nano-sized material of silver, copper, and zinc are potentially source for antimicrobial properties, while inhibit the cell adhesive. The second chapter, is the scope of the present investigation. The presence of silver, gold, and bimetallic ruthenium, cerium oxide, nanoparticle, gloriosa, superful leaf extract in other is evaluated by antibacterial, and antifungal properties in and alginate encapsulated root tip of explant. Investigate the cell division, pollen germination activity of gold nanoparticle, terminal of leaf extract. Investigate the synthesis of terminology of fruit extract and in vivo seed germination activity. So the silver nitrate, gold chloride, ruthenium chloride, cerium trichloride, heptahydrate are used for the precursor. We are using the simple, already well known, the 100 gram of flesh cut leaf and 100 ml of double distilled water, boil at 50 to 60 degrees for 5 minutes. Filter the Waldman filter paper. The filter are 250 ml stored at the room temperature. Further uses for 1 millimolar, all the salt are used for the 5 ml of plant extract. You get the final product. But metal oxide is get the final precipitate. It calcinated to 400 degrees for 2 hours. You get the cerium oxide nanoparticles. It was characterized UV fluorescence, XRD, and AFM time analysis. UV analysis show the surface plasma resonance peak. Next, madam. Next slide. Next slide. Next. Okay. Next. Okay. Surface plasma resonance of the nanoparticle. Silver nanoparticle show the 428 nanoparticle, 425 nanometer, and 538 nanometers, and 550 nanometer surface, which is called collective electron charge oscillating in the metal nanoparticle excited by light. In similar manner of the ruthenium nanoparticle, the lambda max 494 nanometers, and the cerium oxide nanoparticles show the 297 nanometer. Because the cerium oxide nanoparticle generate bulky material of 3.19 electron volt, when it is synthesis of nanoparticle 3.72. By contrast, 0.59 electron volt are increased. Whenever the particle size decrease, the electron band gap decrease. By contrast, in our material get the band gap, the particle size decrease and band gap increase. If we get the increased band gap, you can use for biological application. Is a transition of 3+ to 4+ plays an important role for increase the band gap, the presence of CO2 nanoparticle. Next, the fluorescence property. The fluorescence property of plant extra 471 when the silver nanoparticle sold 530 and 540 for the gold nanoparticle. In order to alloy bimetallic nanoparticles show the 530 and 571 nanometers. In a ruthenium nanoparticle show the 462 nanometer. They are ruthenium N into pi bond transition. And in the metal oxide nanoparticle, it didn't say the fluorescence property is order to photoluminescence property. Is the photoluminescence property of the 8 emission peaks is the NV near band edge emission peaks, blue-green emission respectively. The XRD analysis, XRD is the crystalline nature the identified crystal structure using the syrup formula, you get the 4 different peaks, and you get the average average crystalline size 26.53, 21.87 for gold, 5.50 for bimetallic nanoparticle. In ruthenium nanoparticles show the hexagonal structure, and silver gold bimetallic nanoparticles show the face-centric weak structure, and also the cerium oxide nano show the face-centric weak structure. Micro-Raman analysis. Micro-Raman analysis of 455.27 is C880 vibration for cerium oxide is confirmed by cerium oxide nanoparticle. The XPS, the valency determine the valency in oxidation state. It show that oxygen bond confirm the presence of 3E plus 3D 3 by 2 and 88 2.2 9.21 C 3 plus 3D 5.2S respectively. And stem and EDAX analysis of the Ag nanoparticles. Next slide, ma'am. Next slide. Next. Okay. Stem and EDAX analysis. Same as morphological analysis, surface morphology. EDAX is elemental analysis. The gold nanoparticles show the spherical, gold, silver, bimetallic nanoparticles spherical morphology in the range of 20 to 50 nanometers, and bimetallic show the 10 nanometers is to confirm the EDAX profiles. And another thing is the ruthenium and cerium oxide nanoparticles show the 35 nanometer of spherical nanoparticle, and the cerium oxide nanoparticles show 10 nanoparticle. When you get the below 5 nanoparticle is considered the quantum confinement. When you get the EDAX profiles, confirm the electron volt to confirm the materials. 3 electron, 2.1 electron, 2.2 electron, 2.6 and 4.8 respectively of silver, gold, bimetallic and ruthenium cerium oxide nanoparticles. In addition, some of the peaks of the percentages of the oxygen, sodium, potassium of the A nanoparticle and C and O. It is due to the plant impurity in the presence of the plants of the material. AFM analysis, surface topology. Surface topology analysis without affecting the electron beam. This conductor is a conducting mode or non-conducting mode. Generally, is taken to the non-conducting mode. Is show the 20 to 50 nanometer, 20 to 50 nanometer silver and gold, and bimetallic 10 to 20 nanometers. The ruthenium particles show the 20 and cerium oxide to 10 nanometers. TEM analysis. TEM is accurate particle size was evaluated by TEM analysis. The gold of same manner 20 nanoparticles silver, gold, and the alloy nanoparticles show bimetallic nanoparticles showed 5 nanometers. And TEM analysis is the ruthenium 10 nanometer, cerium oxide 5 nanometer, below 5 nanometer as considered as a quantum confinement. These nanoparticles was investigate the antimicrobial antifungal activity. The Staphylococcus aureus, Streptococcus pneumonia, and so on. So the 2 gram positive on 5 gram negative bacterial and 6 fungal strains used for the antibacterial antifungal activity. It show the silver nanoparticles show the better bimetallic nanoparticles show better antibacterial activity, and followed by the silver nanoparticle and ruthenium nanoparticle. By contrast, the gold nanoparticle cerium oxide nanoparticle didn't show any activity due to the biocompatibility. And the antifungal activity show the better activity of ruthenium nanoparticle, followed by the bimetallic nanoparticle silver gold and silver and cerium oxide nanoparticle. By contrast, gold nanoparticle, the yellow color indicate the gold nanoparticle did not show any activity antibacterial and antifungal activity. Why did not show the antibacterial antifungal activity? So if the negative negates go to the positive impact, we can use for the another application. So why you did what is the exact mechanism of the antimicrobial activity? So the E. coli, the model diagram of E. coli. Next madam. Next slide. The factorial image. Next. Okay. This is the mechanism. The bacterial cell of the gram positive and gram negative. You already know, well known about the gram negative bacteria of the thin layer and positive or thick layer. So is to show the E. coli model as I will explain the antimicrobial bacterial E. coli models. So when you contact with all nanoparticles, generally all nanoparticle positive charge, the bacterial cell membrane is negative charge due to the electrostatic interaction or attraction. It bind with the surface and migrate with the cytosol. Cytosol inactivate the ribosomal subunit, the protein synthesis, DNA replications, and DNA replication. And another one thing is the respirational organ of mesosome. The E. coli, the respirational organ of mesosome bound in the cell wall boundaries in invisibly active the cell division and respiration. So this silver nanoparticle or any other nanoparticle inactivate the mesosomal function and increase the ROH generation. Is hydrogen peroxide is toxic component is easy kill the bacterial survivability. And the electron transport in the surface, in the ADP converted to ATP in the convention appeared in the cell wall. The cell wall damage is lose the electrolyte. The bacterial cell finally dead. The immobilization. Okay. So overall the results show the gold nanoparticle didn't show any activity in order to a positive impact. Already well known the sodium alginate plus calcium chloride gives the calcium alginate beads and sodium chloride. So gold chloride plus MS medium plus 4% of sodium alginate and immersed in the 4% gloriosa root tip explants, you get the 100 millimolar calcium chloride, you get the beads form. With the plant growth regulator, in investigate a regenerative of tissue culture medium, it showed that this is the beads. Madam, previous slide, please. Previous slide. Okay. This is the beads, sodium alginate beads with the root tips explants in inner is introduced in the medium, MS in basal medium, Morashik and Skug medium. Next one, 50%. That means 500 micromolar concentration of gold chloride. Another one is 1000 micromolar molar concentration of gold nanoparticle inca beads of transfer into the medium only show the 1000 micromolar gold nanoparticle 94% of rooted shootlets. All beads are produce a rooted shootlet regeneration, but in the 50 micromillimole only for root only rarely produce the shoot apex. And is 33% of darkish greenish multiple roots are produced. Overall, the plants of hardening, overall the 30 plants are introduced to the hardening purpose. 3 are died, 27 are present. Overall, 80% of people, 80% of plants are survivable. The 100 microliter encapsulators tip glorious super exo positive impact of root or ensol formation. Generation plants were successfully hardening 80% of plants survival under the natural condition and transplantation. Phytosynthesis gold nanoparticles, cell division, and plant tissue culture of glorious alternate encapsulate techniques. In addition to gold nano, also act as a metal regulating protein induce the regeneration efficiency of plant cells. It can be used for other medicinal plant development. The second one is same procedure. We can use the terminally Arjuna leaf and fruit extractor and to parallelly investigate the 10 gram of leaf extract, fruit extractor in the double distilled water boiled at 50 to 60 degree. In addition of gold chloride, you get the gold nanoparticles. Is investigate the cell division, mitotic cell division of Allium cepa and pollen germination of Gloriosa superpa. This is the UV analysis 530 nanometer, 522 nanometer for leaf and fruits. Due to the harm to get the surface plasmoluminescence, XRD peaks confirm the phase centrifugal structure of gold nanoparticles. The AFM was the spherical nanoparticles, and IDAX profile confirm the silver gold metals present in the substrate, and additionally, say aluminum metals present due to the aluminum substrate. The DLS is one of the very good analysis. If you get the positive or negative, positive 30 and minus 30 millivolt, you get the highly stability. The stability nanoparticle, you get the particle size distribution. The overall average size was evaluated. The zeta potential value of the leaf extract get 43 nanometer, highly stable when compared to fruit extract because of 21.9 millivolt is less stable. The thermal particles are fruit particles, spherical nanoparticles. These are investigated gram negative, 2 gram negative and 2 gram positive bacteria didn't show any antimicrobial activity. It is due to the biocompatibility and non-toxic. The similar result was observed in HeLa cell line and E. coli cells. The mitotic cell division is the very important thing. The gold nanoparticles and divided into 100,000 micromolar and Allium root super sample soaking in the 24 hours at intervals 2 days. You are 2 to 3 centimeters suspension with the nanoparticle 4 hours. The microslide were control sample sampling techniques used. The 1 millimolar HCl solution was used for the 40% saffron. Finally, you observed confocal microscopy. The cell division was evaluated. The cell divisions are evaluated is the mitotic cell division gradually increase the concentration. This is beside of the diagrammatically reveal the mechanism. I will show this. This is the cells, the plant cell contain the cytoplasm and protoplasm, cytoplasm and protoplasm. Sorry, sir. Cytoplasm and nucleoplasm. It is covered with the covered with nuclear envelope. The nuclear envelope have the nuclear pore. The nuclear pore size is the 35 nanometer. They will get that below 20 nanometer, easily migrated with the nuclear envelope and enter to the nuclear plasma. After that, it bind with the H2A, H2B protein, and because of the electronegative force, the induce the mitotic cell division, it acts as a metal regulating protein. So whenever increase concentration, the mitotic cell division increase the 29 and 40 and 50 and 71, 41 and 71 percentages. Next slide, ma'am. Next. Pollen grain germination. Why we choose the pollen grain of Gloriosa superpa? The Gloriosa superpa pollen grains of the pollen grains are more major pollen grains of infertile due to the temperature, due to the heavy fall rains. Hand pollination is one of the factor to promote the more seeds and fruits, more seeds. So in order, these Gloriosa superpa pollen grains were evaluated the gold nanoparticle, the same protocol used for the used for the basal medium. The confocal image glows the A is the control. After increase the concentration of gold nanoparticle was increase the pollen germination. It show the gradual increase of gold nanoparticle increase the pollen germination activity as a 2D and the normal microscope image also the same trend, because the pollen grains have survivability more than 1000 years. If you get the old pollen grains, it didn't grow. If you use the gold nanoparticle, you easily generate that 1000 years before plants in haploid plant production. Similarly, Malvia sylvestre and Yucca flamandaceae plants did not show any activity in previous pollen germination attempt. So in order to invention of this gold nanoparticle used for this plant pollen germination, you can get the positive response. Because of the gold nanoparticle is acted as a metal regulating protein and metal regulating proteins. Another seed germination activity. Seed germination activity we can use for the fruit extract. The same tray, the seeds were treated with the sodium hypochlorite and treated with different concentrations. The 100 number of seeds were calculated for seed germination text, 20, 30, 40, 50 days intervals. This is a monoclonal plant. Gloriosa superpa is a monoclonal plant. It's a monoclonal plant seed. The mechanism, the gold nanoparticle and water soaked in the overnight solutions. The imbibition process is activated GA3. GA3 is the gibberellic acid. After the DNA synthesis, DNA to RNA to alpha amylase. Alpha amylase is the only alpha amylase is produced by Aluron cell, only peculiar system in the monoclonal Aluron cell, and the alpha amylase enzyme produced is starch converted to sugar, simple sugar. The simple sugar is easy to germinate the plant seedlings. In the normal condition, plants are 50% survival viability of the Gloriosa superpa, as well as not only germination is increase the biomass. That means of 30 days, 40 days, 50 days plus is the rhizome will be increase the size. It can show this the slide. 54, 74, 93% of increase the concentration with increase the biomass. And the seedling node, internode also increases 7% when compared to control, and leaf initiation also increase as compared to control. Root initiation as well as biomass increasing in the biomass also increasing. The seed germination index show the 50 to 100 millimolar. They enhance the 20%, 20.33%, and 39.67% of seed germination compared to control. Node elongation also found to the same trend of 50 microliters of nanoparticle. Node elongation 4.5 was revealed that control the 4.33 respectively. By contrast, most significant effect node elongation observed 100 millimeter of nanoparticle. The observed phenomena by attributed to increase the level of GA3 due to the response of shoot elongation. The antagonistic impact of A nanoparticle reduced the function of ethylene glass, increased the leaf number of Gloriosa superba, leading the total fresh weight of Gloriosa superba seedling rhizome was increased by the 2.40 and 5.18 times exposed 500 to 100 millimicro nanoparticle compressed with the control. Therefore, the seed germination activity of plant vegetative growth have to depends upon the A nanoparticles. This was published in the industrial product. Next slide, ma'am. Next. Excuse me, ma'am. Next slide.
Sir, I am sharing, sir.
Next
This one, sir.
Next one. Next
Okay,
one. Next one.
This one, sir.
Next one. Next one. Previous one.
This one, sir.
Okay, madam. I will continue. No problem.
Excuse me, sir.
No problem. Previous one. Line number. You go to slide number 83.
Okay, sir. Is this, sir?
Okay.
Excuse me,
This
sir.
invention. Okay, madam. This
Okay, sir.
invention was found in the Vertical News and News RX. The germplasm, the first report of germplasm was reported by the investigation of Alapa University of the green cells of gold nanoparticle using the terminal original leaf extract and enhance the mitotic cell division pollen germination activity. Next slide. The fourth chapter, the green cells of gold nanoparticle using the Philanthus indicus, the Kotangarandai plant used for plant cell and aquatic crustaceans Artemia nephili. Next slide, madam. Next. It shows is leaf extract, generally known as the Philanthus indicus, 10 gram of this one. The UV analysis show the 531 nanometers and same analysis spherical morphology. The XR is high crystalline nature. TEM also the spherical morphology. Next slide, ma'am. The mitotic cell divisions show the increase the concentration of the cell divisions. So 10% of A nanoparticles showed this one. 10% gold nanoparticles showed a high response, 87.66% of mitotic cell divisions. Because of the general cell, total cells were counted by 100 cells. Total number of cell counted, total number of cell divided into 100 is calculating the mitotic index. The prophase are not detected. And controlled total number of cells, prophase 90, metaphase, anaphase, telophase. Generally, we consider the prophase, metaphase, anaphase, telophase. Finally, the percentage was calculated. So, increase the concentration, the gold nanoparticle induce the mitotic cell division. Next, the pollen germination. The pollen germination. The same trend is trend. Is increase the concentration of gold nanoparticle. Next slide, ma'am. It clearly shows the pink color the present in the pollen grains pollen tubes. Next. This also show the Artemia nephili is a crustaceans, is a zooplankton. You can use it for gold nanoparticle. Didn't show any toxicity. You can use the silver and ruthenium and other metal nanoparticle. Is get the 50 to 75% of mortality. Overall, the report eco-friendly techniques of synthesis of Sri Indian leaf extracts and gold nanoparticle is a UV visible FTA XRD and TEM analysis. The 10% dose of metallic cell division pollen germination express the in vitro toxicity against the aquatic pest and Artemia nephili. So gold progressively improve the gold nanoparticle metallic cell division and pollen germination. It is 100% survivability of Artemia nephili. There is no toxicity of cytotoxicity on and non-target organism of Artemia nephili. Next slide, ma'am. Fifth chapter, chitosan nanocrystal used for the potential antimicrobial plant growth promoters. Everyone know the biopolymers. The cellulose is the first polymer. The second most polymer is chitosan, derived from the derived from the shrimp and crabs and fungal material. Is synthesis chitosan nanoparticles show that 250 nanometers is a 5 into 5 chance to confirm the chitosan nanoparticle. It show the XRD analysis. The XRD analysis the well crystalline peaks. The 20 is the carbon based materials. In the crystalline section calculated by syrup formula 6.9 nanometers. It show the 20 to 100 nanometers average size of 50 nanometers is the evaluated antibacterial activity. Didn't show 1 to 1, 3, 5, 10 mg per concentration. 5 and 10 only show the 10% of 10 milligram concentration show the antibacterial activity when compared to positive control of kanamycin. By contrast, the antifungal activity show the very good response. They progressively increase the concentration of chitosan nanoparticles. In the different nanoparticles is a root gel method. It show the Phleranthus indicates plants inoculated into the media. It show that root initiation, shoot initiation, leaf initiation. The 15 and 30 days intervals gradually increase the 10% of 1% of chitosan nanoparticles. The chitosan nanoparticles is very good biocompatibility and biodegradability. It also induces calcium regulating and drought resistant properties. The chitosan positively charged can easily bind with negatively charged bacterial cell peptidoglycan due to the electrostatic interaction that through the enter the microbial cell promote the leakage of intracellular electrolyte and leads to cell death. Transfer the poor fungal cell membrane infecting the host protein as well as intracellular signaling leads to bacterial cell and fungal lysis. The sixth chapter. Next, madam.
Okay.
92.
This one, sir.
Okay. This one, ma'am. The facile synthesis characterization of enhance the biological application of zinc and calcium doped zinc oxide nanoparticles. Will the zinc oxide nanoparticles show the band gap 3.31 when you add the calcium doping 3.23 electron volt? If you go zinc oxide nanoparticles show the very good antimicrobial activity, but calcium doped zinc oxide reduce the antimicrobial activity and enhance the plant tissue culture. This is the theme of this slide. Is further confirmation of zinc is next slide, ma'am. Zinc is one of the reduced micronutrient plant development, playing the key role physiological function, nitrogen metabolism, energy transfer, protein synthesis, plant growth, hormone production, the internode elongation. Consequently, zinc deficiency can alter the plant growth progress, such as accumulation, small leaf formation, flowering, and fruiting. Reduce the seal yield, increasing the oxidative stress. Calcium ions is also essential micronutrients, plays a vital role of development of growing plant cells and cell wall stability. It also shows the actual plant enzyme intercellular communication and cellular activities to promote the cells and tissue elongation, pollen tube germination. This combines the both micronutrition nanomaterials. Surface modification could enable to increasing the bionutrient for plant development. So this is the TG analysis. The aspipet sample was calculated at TG analysis at 350 degree of zinc oxide, pure zinc dope, zinc nanomaterials. Next slide. This is a TG analysis. Thermogravimetric analysis show the zinc oxide nanoparticle, the thermal loss of 320 degrees. When you add the calcium dope nanoparticles, so the higher weight loss. In the UV analysis, the pure zinc oxide nanoparticles show the 370 nanometer. When addition of calcium doping 382 nanometers, the band of value of 3.31, 3.23 electron volt. The XRD patterns clearly show there is no crystalline changes variations. Is confirmed the figure B. Next one, ma'am. The same images also show the zinc oxide nanoparticles or spherical morphology. Is calcium doped zinc oxide nanoparticles or show the look like a ball-like structures is confirmed by the Idhaya spectrum. And the first one is the zinc and oxygen. Second one zinc and calcium and oxygen. Is show the elemental analysis of white and white percentage or atomic percentage. The elemental mapping analysis, the fluorescent property. The black color is all over the material. The red color show the oxygen, and the green color show the zinc oxide nanoparticle, and green with the red show the zinc oxide nanoparticle. B figures show the black image. The overall the calcium doped zinc oxide. Red color reveals the oxygen, and yellow color reveals the calcium, and green color reveals the zinc oxide nanoparticles. Is overall red, yellow, green, or zinc oxide nanoparticles. Is what confirm the calcium metal present in the material. The TEM analysis also show that the spherical morphology, minute structure or spherical structure of 10 to 30 nanometers. By contrast, the zinc doped nanoparticles showed 100 to 400 nanometers. When doping the calcium ion, the increase of particle size is sphere morphology. So these were tested into the antimicrobial activity against the 5 mg against the gram positive and gram negative factors in the Staphylococcus aureus and gram E. coli. In the 5 mg concentration, zinc oxide nanoparticles show the better response as compared to the calcium dope nanoparticle. Because when calcium doping, the decrease of antimicrobial activity. The positive control used for DMSO, negative control used for DMSO, and positive control for rifampicin, 5 mg concentration. It's another application of the different concentration of zinc oxide and calcium doped zinc oxide nanoparticles. So the individual Philanthus indicates the sample inside MS media incorporated the different concentration of zinc oxide and calcium doped zinc oxide, gradually increase the shoot initiation and leaf initiation and node elongation as compared to pure zinc oxide nanoparticles. The calcium doped zinc oxide nanoparticles, the bimetallic nutrient, bimetalloxide nutrient, enhance the shoot initiation as well as leaf shootlets, leaf initiation, node elongation. The zinc oxide and calcium doped hexagonal structure using the X-ray diffraction. The TEM analysis of both are exhibit a spherical shape. The elemental compositions of zinc oxide have determined the Idhaya spectra. They also demonstrate that zinc had a higher antimicrobial activity, both Staphylococcus and E. coli, then calcium doped zinc oxide nanoparticle. Additionally, the both nanoparticle in vitro regeneration Staphylococcus Philanthus indicates shootlets was examined. As expected, that calcium doped zinc oxide nanoparticles are vegetative growth of Philanthus more effectively than zinc oxide nanoparticles due to the synergistic effect. Zinc oxide could not use for can be used for bioimaging application, biomedical, and used for another bionutrient in plant tissue cultures. The overall conclusion: the phytosynthesis of gold, silver, gold, bimetallic, ruthenium, cerium oxide nanoparticles using aqueous leaf extract. To produce the triangular spherical shape of gold, silver with the average size of 20 nanometers, cerium oxide, bimetallic, and ruthenium of 5 nanometers to 10 nanometers. The result significantly effect of the bimetallic nanoparticles, go silver and ruthenium towards antimicrobial activity. Ruthenium showed the inhibit the effect of the fungal activity compared than the bimetallic nanoparticles. The cerium did not show any antibacterial activity. The gold nanoparticle did not show a significant effect in the activity non-toxicity. This suggests the photosynthesis metal oxide nanoparticle beneficially application in the field of bacteriology and pharmacology drug development applications. And 100 micromolar age nanoparticle encapsulated root tip beads of Gloriosa superpa exhibit a positive impact of root or shootlet formation. The 85% of survival under the natural condition, and the photosynthesis biochemical A nanoparticle cell division and in vitro plant tissue of Gloriosa superpa alginate amplification techniques. It's also used for other endangered medicinal plants. The terminal origin of fruit extractors show the 5 to 50 nanometers, average weight of 25 nanometers. Did not show any antibacterial activity. The terminal effect in the mitotic cell division, pollen germination tested in found to be more significant. There is no carcinogenic cytotoxic effect in the cell of pollen grains as well as mitotic cells. The terminal fluid extracted used or treated a Gloriosa seed germination activity. The result confirmed that great potential of induced seed germination have positive impact on the plant vegetative growth of superpa seedlings. The different concentration of the chitosan nanoparticle tested again grow promoters of Silanthes indicate shootlets in vitro. So co-formulation the alginate rooting of root gel. The chitosan promote the growth of shoot or root as well as in initiation of Silanthes indicates 50 to 30 days intervals. Zinc oxide had higher antimicrobial activity of Silanthes indicates. Staphylococcus aureus and E. coli, the calcium doped nanoparticles and the antagonistic effect of the doping process. The calcium doped zinc oxide nanoparticle vegetative growth of Silanthes indica more effective zinc oxide due to the synergetic effect. The green nanomaterial beneficially applicates the field of cell division, pollen germination, seed germination, endangered plant species. Thanking you. Any question and answers? Any questions?
Thank you, sir. I can no other word make but thanks and ever thanks. Thank you so much, sir. I am still struggling to find the right word to properly describe how fantastic content you have presented here. Thank you again, sir. Asking question is the first step one can take towards knowledge. Participants, kindly post your queries here. Sir, shall I convey the queries?
Okay.
Sir, the first one is, what countries are achieving nanotechnology?
Well, the developed countries look like South Korea, China, USA, and European countries. All countries are achieved nanotechnology aspect. India is a developing countries nowadays. The last 10 years back, 10 to 15 years back is new technology is developed. You produce more department in several colleges and universities. It's a new field.
Thank you, sir. The second one is, how are nanomaterials regulated?
Regulator. Some of the nanomaterials are regulators. The gold or silver nanoparticle, some of the zinc nanoparticles, it acts as a metal regulating protein. The zinc 2+ ions gold nanoparticles is open the metal box is promote the cell division, promote the DNA replication, protein synthesis, is enhance the mitotic cell divisions. By contrast, silver nanoparticle is arrest the mitotic cell division, is produce the carcinogenic effect. So zinc and gold nanoparticles are metal regulating protein. Next.
Thank you, sir. What should be the color of zinc oxide nanoparticles?
The zinc oxide depends upon the synthesis protocol. If you synthesis protocol can used for the plant material, you get the yellowish white. When if you get the 100-degree calcination, 100-degree, the room temperature synthesis zinc oxide nanoparticle is get the yellowish white. If you calcinated means the annealing means heated at 350 degrees Celsius, 150 to 350 degrees Celsius, you get the pure white, chalk powder like white color, you get the sample. The sample color change depends upon the bioactive component. If you use for the if you synthesis for the green synthesis manner, you can use the plant extract and so on. So there's so many of tanning component, you get the yellowish color. If you get the chemical synthesis, you get the whitish color, ma'am.
Ma'am, one more question, ma'am. How to identify the biomedical activity of metal oxide and the composite?
Okay, madam. The metal oxides, when composites. Madam, one more time, please, madam.
How to identify The biomedical activity of metal oxide and composite, sir.
Okay, madam. Metal, if active bio, one minute, please. Metal composite is more than 2 metals if you used for carrier because of the activity. If you give the directly metal oxide nanoparticle, didn't show any activity. If you need to nanocomposite. Nanocomposite means chitosan. Chitosan is look like a partially positive or negative. Okay, generally positive. Chitosan, graphene oxide, and metal oxide. This is a combination. You holding capacity of drug carrier. If you get the very unique material, is progressively minimum effect. If you used for nanocomposite, is get the beneficial application is 100 more than 90 to 100% efficiency, ma'am, when compared to metal oxide. The nanocomposite is a very good drug carrier.
Okay, sir. Okay, sir.
Thank you so much, sir.
Okay, madam.
Shall I validate, sir?
Okay, madam.
Gratitude
Now-
opens the door to the power of wisdom, the creativity of the universe. I thank our God Almighty who showered His blessing in occasions. The thank of our principal, Reverend Sister Eugene Amala, who has been supporting continuously for us. I thank of our college secretary, Reverend Sister Claudina Arokia Mary, our vice principal, Reverend Sister Vimali Mary, and our administrator, Dr. Andoni Ammal, who has been supporting in various ways. The thank of resource person, Dr. Kasi Gopinath, for his inspiring speech on recent trends in green nanomaterials used for plant development and biomedical applications. Thank you, sir. Thank you very much.
Thank you, ma'am.
I consider it's a great privilege to purpose of vote of thanks to all participants for your cooperations in making this FTP as a memorable success. Once again, I thank you all, one and all.